Alright team, let's get into it. Today we're tackling a fundamental concept in medicine, something that underpins so much of what we do as healthcare professionals: antigens. You've probably heard the term thrown around a lot, especially in immunology, infectious diseases, and even cancer therapy. But what exactly is an antigen? Put simply, an antigen is any substance that prompts your immune system to produce antibodies against it. Think of it as a molecular red flag. When your body sees something it doesn't recognize, this substance, the antigen, triggers an alarm. This alarm is what we call an immune response, a complex and coordinated defense mechanism designed to protect you from harm. Understanding antigens is not just academic, it's incredibly practical. It's the key to understanding how vaccines work, why we have allergies, how our bodies fight off infections from bacteria and viruses, and why organ transplants can sometimes be rejected. It's also at the heart of autoimmune diseases, where the body mistakenly targets its own cells. Every time you administer a vaccine, treat an allergic reaction, or manage an autoimmune condition, you're dealing with the consequences of an antigen-antibody interaction. So, grasping this concept early on in your medical or nursing journey will provide a solid foundation for countless clinical scenarios you'll face in the future. The word antigen itself is a portmanteau, a blend of antibody generator. This name perfectly captures its primary function. These substances can be proteins, carbohydrates, or other molecules found on the surface of pathogens like viruses and bacteria, or even on foreign materials like pollen or transplanted organs. When these molecules enter your system, your body's surveillance team, the immune cells, gets to work. They identify the antigen as foreign and initiate a cascade of events to neutralize the threat. This response is highly specific, meaning the antibodies produced will only target the specific antigen that triggered them, much like a key fitting into a specific lock. So, as we dive deeper into this topic, remember this core idea: an antigen is a trigger. It's the wanted poster that the immune system uses to identify and hunt down invaders. We're going to break down the different types of antigens, what they're made of, and how your body brilliantly uses them to distinguish friend from foe. This knowledge is your first step towards mastering immunology and becoming a more effective clinician. Pay close attention, because what you learn here will stick with you for your entire career. Let's begin this journey and unlock the secrets of these fascinating molecules. Now that we have a basic idea of what antigens are, let's talk about a crucial distinction your immune system has to make every single second of every day: the difference between self and non-self. Your body is full of its own molecules and cells, all of which have substances on their surfaces that could technically act as antigens. These are called autoantigens, or self-antigens. Under normal, healthy circumstances, your immune system has been trained from a very early stage to recognize these autoantigens as part of you. It learns to ignore them through a process called immune tolerance. This is a vital safety mechanism that prevents your immune system from attacking your own tissues. On the other hand, we have foreign antigens, which are also known as non-self antigens. As the name suggests, these are molecules that originate from outside your body. They are the classic invaders. This category includes a vast array of substances, from the proteins on the surface of a flu virus or a bacterium like Streptococcus, to the allergens in pollen or peanuts, to the cells of a transplanted kidney from another person. When your immune system encounters these foreign antigens, it correctly identifies them as a potential threat. This identification is the first step in launching a powerful and targeted defensive response to eliminate the invader and protect your body from disease. The breakdown of that self-tolerance mechanism I mentioned earlier is what leads to autoimmune diseases. In conditions like rheumatoid arthritis, lupus, or type 1 diabetes, the immune system loses its ability to distinguish self from non-self. It mistakenly identifies autoantigens, like proteins in the joints, skin, or pancreas, as foreign threats. This leads to a devastating friendly fire incident where the body's own defense system attacks and destroys healthy tissues. Understanding this distinction is therefore critical for diagnosing and managing these complex and often debilitating conditions that affect millions of people worldwide. So, to recap, your body is constantly scanning for antigens. It maintains a sophisticated do-not-attack list for all your own autoantigens, ensuring cellular harmony. But when it detects a foreign antigen, it switches into attack mode. This elegant but powerful system of recognition is the cornerstone of a healthy immune response. The balance between tolerance to self-antigens and reactivity to foreign ones is what keeps you healthy. In the next sections, we'll focus primarily on the foreign antigens, as they are the ones that provoke the classic immune responses we study in medicine. Let's zero in on the foreign antigens, the non-self molecules that our immune system is primed to attack. These are the main players in the story of infection and immunity. It's important to understand that they come in many shapes and sizes from a wide variety of sources. The most common examples we encounter in medicine come from pathogens. For instance, viruses are essentially packages of genetic material wrapped in a protein coat. The proteins on this coat, like the spike protein on the surface of the SARS-CoV-2 virus, are potent antigens that our immune system learns to recognize and target. Bacteria are another major source of foreign antigens. These single-celled organisms have complex structures with numerous potential antigens on their surfaces. The cell wall of a bacterium, its flagella, the whip-like tails used for movement, and the toxins it might release are all packed with molecules that your immune system can identify as foreign. A classic example is the lipopolysaccharide, or LPS, found in the outer membrane of gram-negative bacteria, which is a powerful trigger for a strong immune response. Even fungi and parasites present their own unique sets of antigens that our bodies must contend with. Beyond infectious agents, we encounter foreign antigens in other contexts. Allergens are a perfect example. Substances like pollen, dust mites, pet dander, or certain food proteins are harmless to most people. However, in an individual with allergies, the immune system mistakenly identifies these substances as dangerous foreign antigens. This triggers an allergic reaction, releasing chemicals like histamine that cause the familiar symptoms of sneezing, itching, and swelling. This is essentially an overreaction of the immune system to a perceived but non-existent threat. Finally, in the context of clinical medicine, we must consider antigens from other humans. When a patient receives a blood transfusion or an organ transplant, the cells from the donor contain antigens that are foreign to the recipient's body. The most well-known are the A, B, and Rh antigens on red blood cells, which dictate blood type compatibility. Similarly, transplanted organs have surface proteins, which we will discuss later as MHC molecules, that can trigger a powerful rejection response if they are not closely matched. This is why tissue typing and immunosuppressive drugs are so critical in transplant medicine. To get even more specific, we can classify foreign antigens into two main categories based on where they come from and how they are processed by the body. This distinction is crucial because it determines which arm of the immune system will be primarily responsible for dealing with the threat. The two types are exogenous antigens and endogenous antigens. This might sound complicated, but the logic is actually quite straightforward. Let's break it down. Exogenous antigens, from the Greek exo, meaning outside, are antigens that have entered the body from the external environment. Think of them as invaders that are currently outside your cells, perhaps floating in your bloodstream or tissues. This category includes most bacteria, free-floating viruses that haven't yet infected a cell, pollen, dust, and parasites. These antigens are taken up from the outside by specialized immune cells called Antigen-Presenting cells, or APCs, which we will discuss in detail shortly. These APCs essentially eat the foreign substance, break it down, and then display a piece of it on their surface to alert other immune cells. Endogenous antigens, from the Greek endo, meaning inside on the other hand, are generated from within one of your own body cells. This happens when one of your cells becomes compromised, either because it has been infected by a virus or because it has become cancerous. When a virus infects a cell, it hijacks the cell's machinery to produce new viral proteins. These viral proteins are endogenous because they are made inside the cell. Similarly, cancer cells produce abnormal proteins due to genetic mutations. These abnormal proteins are also endogenous antigens that signal to the immune system that the cell is damaged and needs to be destroyed. This distinction is the key to understanding how your immune system mounts a tailored response. The pathway for presenting exogenous antigens primarily activates helper T cells, which then orchestrate the overall immune defense, including stimulating B cells to produce antibodies. Antibodies are great for targeting invaders that are outside of cells. In contrast, the pathway for presenting endogenous antigens activates cytotoxic T cells, also known as killer T cells. These cells are specialized assassins that directly seek out and destroy your body's own infected or cancerous cells, eliminating the source of the problem from within. So what are these antigens actually made of at a molecular level? While technically any substance can be an antigen, not all are created equal in their ability to provoke an immune response. The most potent antigens are typically large, complex molecules. The immune system is much better at recognizing and responding to something big and intricate than something small and simple. The chemical nature of the substance plays a huge role in its antigenicity, which is its ability to bind with antibodies or T-cell receptors, and its immunogenicity, which is its ability to trigger a full-blown immune response. Proteins are by far the most common and powerful antigens. This is because they are large molecules made up of long chains of amino acids, folded into complex three-dimensional shapes. These intricate shapes provide a wide variety of unique surface features that the immune system can recognize. Most of the antigens we've discussed so far, viral coat proteins, bacterial surface proteins, and abnormal cancer proteins, are all protein-based. Their complexity and structural diversity make them excellent targets for a highly specific immune attack. Polysaccharides, which are large carbohydrate molecules, can also be potent antigens. A good example is the polysaccharide capsule that surrounds certain types of bacteria, like Streptococcus pneumonia. These capsules can act as antigens, and are often the target of vaccines designed to protect against these bacteria. Lipids and nucleic acids like DNA and RNA on their own are generally poor antigens because they are relatively simple and structurally repetitive. However, when they are combined with proteins or polysaccharides, forming molecules like lipoproteins or glycoproteins, their immunogenicity increases dramatically. The protein or carbohydrate part acts as a carrier, helping to present the lipid or nucleic acid to the immune system. It's also important to know about a concept called haptens. A hapten is a small molecule that is antigenic but not immunogenic on its own. This means it can be recognized by an antibody, but it's too small and simple to trigger an immune response by itself. However, when a hapten binds to a larger carrier protein in the body, the hapten carrier complex becomes immunogenic. A classic example of this is the drug penicillin. Penicillin itself is a hapten, but in some individuals it can bind to proteins in the blood. This new complex is then recognized as foreign, triggering an allergic reaction. This explains why some drug allergies develop. When our bodies face threats from antigens, especially those that hide inside our own cells, what we call endogenous antigens, they call in the special forces. This branch of the immune system is known as cell-mediated immunity. Unlike the humoral immunity branch, which relies on antibodies floating freely in the blood and other bodily fluids, cell-mediated immunity involves direct cell-to-cell combat. Its primary soldiers are highly specialized lymphocytes called T cells. This system is actually our main line of defense against viruses, some bacteria that live inside cells like Mycobacterium tuberculosis, fungi, and cancer cells. The development of cell-mediated immunity begins in the bone marrow, where all blood cells, including lymphocytes, are born. The precursor T cells, not yet mature, then travel to a small organ in the chest called the thymus. The T in T cell actually stands for thymus. Inside the thymus, these young T cells undergo a rigorous education and selection process, often called thymic selection. It's kind of like a boot camp for T cells. They're tested to ensure they can recognize foreign antigens, but, crucially, that they do not react against the body's own self-antigens. It's a tough school. More than 95% of T cells don't make the cut and are eliminated. The T cells that graduate from the thymus are now considered naive but mature, which means they're fully functional but haven't yet encountered the specific antigen they're programmed to recognize. They circulate throughout the body, patrolling the blood, lymph nodes, and spleen, constantly on the lookout for trouble. When a naive T cell finally meets its specific antigen, properly presented by an antigen-presenting cell, it becomes activated. This activation is the trigger that transforms the T cell from a passive patrol unit into an active-duty soldier, ready for battle. Once activated, the T cell rapidly multiplies, creating an army of clones all programmed to recognize the same specific antigen. These clones then differentiate into two main types of effector cells. The first are the cytotoxic T cells, also called CD8+ T cells, the direct killers. They hunt down and destroy infected or cancerous body cells by releasing toxic chemicals that punch holes in the target cell's membrane. The second type are the helper T cells, or CD4+ T cells, the battlefield commanders. They don't kill cells directly, but but instead release chemical signals called cytokines to coordinate and amplify the entire immune response, activating other cells like macrophages and B cells. For cell-mediated immunity to work, the T cells need to be shown what to attack. They can't just recognize a free-floating virus or bacterium on their own. They need a messenger, a special type of cell that processes the antigen and presents it to them in a very specific way. These messengers are collectively known as Antigen-Presenting cells, or APCs. Their job is absolutely critical. Without them, the T cell response would never get started. There are three main types of professional APCs we need to know: macrophages, dendritic cells, and B lymphocytes. First up are the macrophages. The name literally means big eater, which is a perfect description of what they do. Macrophages are phagocytes, meaning they engulf and digest cellular debris, microbes, and anything else that doesn't belong. They're found in virtually every tissue of the body, acting as a combination of garbage collector and security guard. When a macrophage engulfs a pathogen, it breaks it down into smaller pieces, antigens, inside a vesicle. It then takes these antigen fragments and displays them on its cell surface, carrying the message to a helper T cell to signal that an invasion is underway. Next we have the dendritic cells. These are the most potent and effective APCs in the business. They're considered the master activators of naive T cells. Like macrophages, they're found in tissues that have contact with the external environment, such as the skin, where they're called Langerhans cells, and the lining of the nose, lungs, and intestines. Their defining feature is their long, branching projections, which look like the dendrites of a neuron. This shape gives them a massive surface area to interact with other cells. When a dendritic cell captures an antigen, it migrates to the nearest lymph node to present it to the T cells residing there, initiating a powerful and targeted immune response. Finally, we have the B lymphocytes, or B cells. While B cells are primarily known for producing antibodies as part of the humoral immune response, they also play a key role as APCs. A B cell has a unique B cell receptor on its surface that's highly specific for one particular antigen. When it encounters that antigen, it can bind to it and internalize it. Just like macrophages and dendritic cells, the B cell then processes the antigen and presents fragments on its surface. This allows it to interact with a helper T cell that recognizes the same antigen, a crucial step for the B cell to get the permission it needs to become fully activated and start producing antibodies. So how exactly do APCs present an antigen? They don't just hold it out randomly. They use a specialized set of protein molecules on their cell surface called the Major Histocompatibility Complex, or MHC. You can think of MHC molecules as molecular display cases or ID card holders. Their job is to bind to antigen fragments and hold them up on the cell surface so that T cells can inspect them. This MHC antigen combination is the precise signal that a T cell receptor is designed to recognize. In humans, the genes for MHC proteins are referred to as the human leukocyte antigen, or HLA system. This is why you hear about HLA typing for organ transplants. It's all about matching these critical MHC molecules. There are two main classes of MHC molecules that you absolutely must know: MHC class 1 and MHC class 2. They have different structures, are found on different types of cells, and present different types of antigens, leading to very different immune outcomes. It's a beautifully logical system. MHC class 1 molecules are found on the surface of almost all nucleated cells in your body. That means basically every cell except red blood cells. Their job is to present endogenous antigens, those proteins made from within the cell. This is the system for flagging an internal problem. If a cell is infected with a virus, it will chop up some of the viral proteins and display those fragments on its MHC class 1 molecules. Similarly, if a cell becomes cancerous and produces abnormal proteins, it will display those on its MHC class 1 molecules. This display acts as a distress signal, essentially shouting, "I'm compromised on the inside, kill me." This signal is recognized specifically by cytotoxic T cells, or CD8+ T cells, which then carry out their duty to eliminate the damaged cell before the problem can spread. It's the body's internal surveillance system. In contrast, MHC class 2 molecules are found only on the surface of professional antigen-presenting cells, macrophages, dendritic cells, and B cells. Their job is to present exogenous antigens, those that have been captured from outside the cell. When an APC engulfs a bacterium, it breaks it down and displays the bacterial fragments on its MHC class 2 molecules. This display acts as an alert to the wider immune system saying, "I found an external invader, we need to mount a response." This signal is recognized specifically by helper T cells, or CD4+ T cells, which then become activated and begin to coordinate the overall immune attack. We've covered all the individual players. Now let's put it all together. How does this sequence of events lead to the activation of a helper T cell, the master coordinator of the immune response? It's a multi-step process that requires a series of precise interactions ensuring the immune system doesn't get activated by mistake. Let's walk through the activation of a naive helper T cell, a CD4+ T cell, by an antigen-presenting cell like a dendritic cell. This is a pivotal moment in any adaptive immune response. First, the APC must encounter and process an exogenous antigen. Imagine a dendritic cell in the skin encountering a bacterium. It engulfs the bacterium through phagocytosis and breaks it down into small peptide fragments. These fragments are then loaded onto MHC class II molecules inside the cell. The APC, now armed with the antigen, travels to a nearby lymph node, which is a hub where naive T cells congregate. Here the APC begins to scan the crowd, searching for a helper T cell with a T cell receptor that perfectly matches the specific antigen fragment it is displaying on its MHC class II molecule. This initial binding event is signal 1. The T cell receptor on the helper T cell physically binds to the antigen MHC class II complex on the APC. This is the specificity step. If the TCR isn't the right shape, no binding occurs. However, this signal alone is not enough to activate the T cell. This is a crucial safety measure. To prevent an inappropriate immune response, a second confirmation signal is required. This is known as the co-stimulatory signal, or signal 2. It involves another set of proteins on the surfaces of both cells. The B7 protein on the APC must bind to the CD28 protein on the helper T cell. Think of it as a two-key system to launch a missile. You need both keys turned simultaneously for activation to occur. This two-signal requirement ensures that T cells are only activated by professional APCs that have genuinely encountered a pathogen, since pathogen detection upregulates the B7 protein. Once both signal 1 and signal 2 are successfully delivered, the helper T cell is fully activated. It begins to produce a cytokine called interleukin-2 or IL-2, and also expresses more IL-2 receptors on its own surface. This IL-2 acts on the T cell itself, driving it to proliferate rapidly, creating an army of thousands of identical clones, all ready to fight the specific invader. These newly activated helper T cells are now the conductors of the immune orchestra. They can leave the lymph node and travel to the site of infection to help activate macrophages, making them more effective killers. They can also help activate B cells, authorizing them to produce antibodies against the pathogen. And they can help in the activation of cytotoxic T cells. This carefully controlled activation sequence is a masterpiece of biological engineering, ensuring a powerful response is only unleashed when absolutely necessary. Mastering this pathway is key to understanding both immunity and immunopathology. I know that was a lot to take in, but breaking down these complex processes is what being a great medical professional is all about. If you want to keep building on this knowledge with more clear, simple breakdowns of medical topics, make sure you check out the content we're putting out at Joen Academy. We're dedicated to helping you succeed on your journey. Keep up the great work.